The topic for this session is the discussion of central tolerance, which basically should imply to you that your body needs to tolerate itself and not regard your own tissues as foreign. Because if you remember, the entire premise of the immune response is that these are a bunch of cells and proteins and mediators whose job it is to recognize when there has been an invasion. So the first part of recognizing an invader, of course, is to recognize what you look like. The recognition of self becomes the primary component. Now, central tolerance should imply to you that this is going to happen in the central immunologic organs. We've already spent some time in the bone marrow, and remember that B lymphocytes will carry out all of their developmental stages before they are challenged with antigen in the bone marrow. T lymphocytes will actually leave the bone marrow and go to the thymus, and their selection will happen in that location. Now, in the absence of any foreign antigenic stimulus, those cells are undergoing their normal VDJ recombination. And I think it should make sense to you that because of the fact that this process is so random and so potentially fraught with errors, that there are times when cells are created that have a receptor that would actually be bad for the human host in which they live. And the case where this would happen would be if a receptor were formed that had a three-dimensional complementarity for one of the cells or proteins or components of your own tissues. So if this happens when B lymphocytes are undergoing their development, what will happen is that any of those receptors that bind too strongly to stromal constituents, the stromal cells in the bone marrow, would be induced to undergo apoptosis in that location so that it would not be allowed to leave the bone marrow and potentially attack your own tissues elsewhere. The process for development of central tolerance for T lymphocytes is even more rigorous than this because there is some slippage within the B cell compartment where some cells are actually released which might have a potential for binding to something that you own, but would not be able to do so in the absence of help. But remember, the design of the system is such that the general of the immunologic army is the T cell. And so if that cell does not recognize extremely carefully what is self versus what is non-self, the problem could have catastrophic outcomes. Now, if in the thymus a T cell develops a receptor which actually binds to one's own tissue constituents, then it will be induced to undergo apoptosis in that area. And so the process is really tremendously inefficient for what you would think your biological systems would develop. If you notice, somewhere between 1 and 5% of all T cell precursors are destined to die in the thymus as they go through this process. So it seems very biologically wasteful, but when you think about the importance of the ability of your body to recognize you versus not you, it makes a little bit more sense. Now, the team uniform that tells your immune response who is called the major histocompatibility complex. And that set of initials applies in general to any of those molecules in any species. Specifically, in the human, these are called the human leukocyte antigens, or HLA. And these receptors on the surface of membranes belong to that same family of genes that we've talked about before, which encode the T cell receptor and the immunoglobulin genes. The purpose of these molecules, however, is to present peptides that are being created inside or outside cells in your body and present them to the T cell receptor that we spoke about developing in a previous discussion. So realize that the categories of HLA or MHC, depending on how you would prefer to call them, the categories of those molecules have evolved for different purposes, as you'll see in a moment, but both of them serve as presentation molecules on our antigen-presenting cells to show who is wearing the team uniform and who is not. So, for example, a normal cell of your body would be wearing a normal MHC molecule carrying a normal peptide and would not be recognized as being foreign. But if an abnormal or extraneous protein is being carried, that is going to elicit a response downstream in the cell that is going to collaborate with that APC. Now, as you notice, we have some molecules here that I've mentioned before on the surface of T cells, namely CD4 and 8, which serve as co-receptors for one or the other of these MHC molecules. CD4 is found beside the T cell receptor on cells that recognize MHC class II, and CD8 is found beside the T cell receptor on cells that express MHC class I. So let's talk for a moment about where these molecules are actually found and how they are constituted. If you notice in the table, we have the names of gene products listed here for either the class I or the class II products. In humans, they would be HLA A, B, or C in the case of MHC class I, or DP, DQ, DR in the case of MHC class II. The MHC class I molecule is found on every single nucleated cell of your body. The only cell of your body that does not have class I MHC is a red blood cell. And so that molecule cannot serve for recognition in that case. But remember, red blood cells are extremely specialized cells who have to move smoothly and effortlessly through the body to carry oxygen, etc. So I don't want them to be wearing the uniform that might cause them to be identified as anything unusual. My antigen-presenting cells, which is quite a long list of cells, it could be my macrophages, it could be dendritic cells, it could be B lymphocytes, it could be activated T lymphocytes, and even endothelial cells. These will possess class II MHC, which is going to serve to send the signal that the cell wearing that product is asking for help in an immune response. Notice that both of these molecules are expressed codominantly, and this is the most polymorphic genetic system in the entire human being. Therefore, what I'm saying is there's a tremendous amount of diversity within the system itself. And if each of your cells who has a particular category of MHC is wearing a copy from your mother and a copy from your father, notice that things are going to get quite complicated quite quickly. If I have got two molecules of DP, DQ, and DR, one from my father and one from my mother, as well as in many cases, two copies of A and B and C on each of my cells. Realize that if I were to talk about a complete match between two human beings, I would have to be matching 6 of class 2 and 6 of class 1 to say that the two individuals had a perfect match. And if there are thousands of these in different ramifications, I think you can realize why it is when we speak later about transplantation that it becomes such a complex issue to find two human beings who are not genetically related to one another who actually have similar HLA haplotypes. Now, when we look at the molecule itself, notice that class 1 has a long alpha chain and beta-2 microglobulin. The alpha chain is going to have a groove, which is going to accommodate a peptide. Beta-2 microglobulin is not even encoded on the same chromosome as the rest of the molecule. And its purpose is simply to hold that alpha chain in the configuration it needs to so that that groove is formed. The class 2 molecule has 2 chains, and once again you see we have fallen into the Greek alphabet to simply name these. These have nothing to do with the alphas and the betas we've spoken about previously. It just became convenient when we named things to name the first one we found, the first letter of the Greek alphabet, etc. Okay? I spoke a moment ago about the co-receptors for the two being CD8 for MHC class 1, CD4 for MHC class 2, and notice, I suspect most of you are way past having problems with this, but notice if you have a total brain cramp and you can't remember which one goes with which, you can think of the fact that MHC class 2 times the CD4 number gives me 8, and MHC class 1 times the CD number also gives me 8. So the product of those two, this is a meaningless memory tool other than to make sure you don't forget. Class 2 responding cells use CD4 as the corresponder, and class 1 recognizing cells use CD8. That is an important identifier in medicine that we will speak about clinically later on. Now, the thymus, which is your second primary immunologic organ, or your second generative immunologic organ, is a bilobed structure in the thorax. And what most people don't realize, because most physicians have never seen a thymus, is that it is a massive organ in the chest of a child. It's actually larger than the heart and the lungs combined. And I think the fact that it is inside the thoracic cavity should cause you to think evolutionarily about the importance of this organ. The thymus, which is your second primary immunologic organ or generative immunologic organ, is a huge organ in the thoracic cavity of a newborn, but by the time an individual reaches adolescence, it has reverted to a fibrous scar. Now, when you think about the fact that the thymus is found inside the thoracic cavity, and you think about evolutionarily how most of our most important organs are indeed found inside a bony casing, I think you can realize that it was a foolish operation in the past to think about removing the thymus if it was abnormally large in the view of the medical community. But horrifying as this sounds, it was indeed common practice prior to 1960 to irradiate the chest cavity of children for whom they felt the thymus was too large and might impede the normal function of the heart and lungs. We now know, of course, that it is the master immunological organ, and so you can anticipate what the problems would have been in children who had this radiation therapy, and it was not a good story. But suffice it to say for now, the thymus is going to be the location where all of the non-self recognition of your T cell components is going to happen. And do remember, of course, that we're speaking only about adaptive lymphocytes here, because there are a whole other category of innate lymphocytes who do not have the same antigen receptors and therefore do not go through these selection processes. Now, the thymus is still visible as a scar in the chest cavity as you look radiographically, and it will be in the superior mediastinum as a faint haze in an adult. But remember, the fact that it regresses by adolescence tells you that the important functions of this organ happen in the really very early postnatal period, when the child is exercising its experimentation with the environment and learning what things are going to challenge it immunologically and what things would best be ignored. Now, as your T cell progenitors leave the bone marrow, they come into the thymus, into the medulla through high endothelial venules, but they move immediately to the cortex, and they will start out as they are still rearranging their VDJ gene segments to make their T cell receptor. They will start out expressing neither of the co-stimulatory molecules on their surface. So we would say then that they are double negative cells, double negative meaning neither CD4 nor CD8 expressed. They complete the formation of the T cell receptor and immediately become double positive, wearing both CD4 and CD8. And now they are capable, because of the T cell receptor, to interact with epithelial cells in the cortex of the thymus. Those are going to express both MHC class 1 and MHC class 2 molecules native to that individual. And if the thymocytes get signaling of an appropriate type from that, they will go on to progress through their developmental stages. But in the absence of signaling, they will simply die by neglect in the area because eventually, as we said, the thymus is going to involute with age. The positive reaction of recognizing one of those two MHC types is going to cause the development of cytokine production, and those cytokines are going to induce those cells that responded to clone themselves and be pushed toward the inner portion of the thymic cortex. They now become single positive cells, wearing only one or the other of the CD markers. Again, as we said, retaining CD8 if they recognized MHC class 1 appropriately, or wearing CD4 if they recognized MHC class 2 appropriately. Now, as they are pushed into the medulla of the thymus, they now interact with medullary thymic epithelial cells, and those cells have specially evolved to evaluate the strength of the binding between the MHC molecule and the T cell receptor. So they will then deliver appropriate signals to the cells that will cause them to develop down one of several different pathways. And I should mention that we now know that the ability of these thymic medullary epithelial cells to express all of the potential MHC class 1 and 2 molecules with peptides that are possible in your entire body is controlled by transcription regulators and transcription factors. AIR is one that stands for autoimmune regulatory factor, and then FESF2, which regulates transcription. And in the absence of those two molecules, realize the medullary cells cannot express all of the molecules from your body, and you will develop a less healthy central immunologic tolerance. Now, if you look at this in a diagrammatic fashion, realize that where we were before was discussing the development of the lymphoid progenitor, and now that cell, destined to become a T cell precursor, will enter the thymus through the medulla, but the beginning of the testing occurs when they reach the cortex. They are initially double negative, but quickly become double positive as they achieve the ability to complete their T cell receptor, and the evaluation of whether that T cell receptor is complementary to class 1 or class 2 is going to cause them to then evolve further down the pathway. In the absence of recognition, they simply die in the thymus as it involutes with age. But in the presence of a strong affinity between the two, I will achieve positive selection by donation of cytokines into the area that will cause the cells to clone themselves. And they will now differentiate into single positive cells with their ability either to recognize MHC class II or, contrastingly, their ability to recognize MHC class I. Those single positive CD4 or CD8 positive cells will now pass into the thymic medulla, where the medullary thymic epithelial cells will deliver the signals that will evaluate the strength of binding between the two MHCs and the TCR of the respective cells. So notice that if there is very strong binding between the T cell receptor and one of the MHC forms, then that's going to result in the fact that I would induce apoptosis in that cell, because it's basically saying that that cell has a T cell receptor that strongly binds to my own tissue types and therefore could cause problems downstream. Those that have intermediate strength of binding or affinity are induced to become regulatory cells of two different types, either those that regulate the helper pathways on the right here, or those that regulate the cytotoxic pathways. And those that have a relatively weak interaction, clear recognition but not tight binding, those would be determined to turn into the final populations of fully effective either helper cells or cytotoxic cells who will emerge from the thymus as single positive CD4 or CD8 cells respectively. So the process is extremely diligent, and as you notice, it's true that a very small number of cells actually survive this process. It is at its height in the immediate period after birth and in young childhood, and at the point that you get to be an old crone like me, or even old crones like most of you, past adolescence, the thymus is no longer doing nearly anything compared to what it started out doing before. Now, when we look at this in summary and compare our discussion of what T cells and B cells do differently, realize that they both evolve from that stem cell in the bone marrow, but then they follow very different pathways as time goes on. The pro-B cell is the cell on the B lineage who has not as yet undergone VDJ recombination and therefore has no portions of receptors even being formed in its cytoplasm. That is true also of the pro-T cell, which by this point is headed for the thymus to undergo its remaining stages of development. The B cell next manages to rearrange the VDJs for its mu heavy chains and begins to express those in the cytoplasm. That stage, the pre-T of the T cell lineage, would have the beta chain formulated and expressed on the surface along with CD3, but it still would not have either CD4 or CD8, and notice that its T cell receptor is not a normal functional one. It has CD3 linked to the beta chain. It has not yet completed its alpha chain. Next, we would move into the stage for the B cell, where we spoke about an immature B having the entire molecule of IgM completed and expressed on the surface membrane. And in the T cell lineage, which is analogous, I would have a double positive cell that had a T cell receptor, and we simply call that a double positive thymocyte. You can say it's immature, but the terminology is usually in the context of how many CD molecules it has on its surface. As we move on, the mature, naive B lymphocyte on the left-hand side by alternative RNA splicing, manages to complete the molecule of IgD. Remember, it has to have the same idiotype, but now I have two different isotypes being expressed on the surface of the cell. Okay? And the thymus is now going through its selection to judge whether its affinity for the MHC or MHC1 or MHC2 molecule is actually appropriately strong. So what emerges from the thymus is a cell with either CD4 or CD8 and affinity for MHC class 2 that is appropriate or MHC class 1 respectively. Now, a couple other things in the center here that I wanted to discuss as universal attributes of cells in both of these pathways is notice that the RAG gene expression and the function of TDT are really only expressed in the earliest of cells, and those cells should never be allowed to be free in the circulation. They should be found in the primary lymphoid organs undergoing this process of development, but they should not be released into the circulation. These are markers for very early cells. Notice it is not until the point of maturity that a lymphocyte can actually recognize antigen or foreign substance in an appropriate way. And as a general rule, if I expose immature cells to antigen, they will be tolerized by those materials, where it is only the mature ones who see a molecule that will make an immune response against it. So your body is sort of hardwired for that system. Seeing your own tissues as you develop is a tolerizing signal. It's not until you leave these organs that you find the cells that are capable of making an active immune response to these materials. Now, before we leave this final portion of the chapter, I wanted to once again do another Make Me Care session here. So remember, the way it works is that I'm going to pose a question. You're then going to pause your video and try to answer these questions for yourself. And then when you're ready, unpause the video and I will give you the answer to the question. So my first Make Me Care quotient is, if a patient presents with circulating lymphocytes with RAG and TDT expression, what can you infer? Pause the video and come back when you're ready. Well, as I just said, both the RAG genes being active and TDT being expressed are functions of immature lymphocytes. So if I have a condition where these are being pushed out of the primary lymphoid organs before they have completed their developmental stages, it implies that I've got crowding in the primary lymphoid organs and the potential of some sort of malignant transformation. So realize these are markers that we use clinically for detection of ALL, acute lymphoblastic leukemia. And so if you think about realizing the stages of development of these cells, you could easily recognize what were cells that were not supposed to be in the circulation. Second, in cases where malignant transformation occurs in a lymphoid cell lineage, immunophenotyping is performed to determine the developmental stage of the involved cells. How would this information be useful to the clinical management team? Pause your video and think about this for a moment and then come back. Well, obviously, I'm not talking about those things that would be markers for use with pharmacotherapy, because obviously we are evolving our knowledge very quickly in how to use markers on the surface of cancers to target them with therapeutic drugs. But what I'm asking here is, how would the developmental stage of a lymphocyte make a difference to how one considers what needs to be done? And the thing that you should think about there is that the more primitive the cell within the lineage, the more mitoses it potentially still has in its lifespan. So it tends to be true that the very least developmentally mature cells that are making up a malignancy in a particular patient are going to be more aggressive and have more growth potential and need to be treated somewhat differently. If I have a very highly evolved cell that has undergone malignant transformation, it has fewer mitoses left in its lifespan, and therefore it might be easier to treat in the long run. Number 3, the thymus is one of 5 immunoprivileged sites in the body. The others are brain and retina, testis, placenta. Okay? Why would you think that a specialized barrier between the organ and the blood would be necessary for normal thymic function? Pause your video and think about that for a moment. Well, the specialized blood to organ barrier systems are those that either are designed to perhaps stop complex molecules from getting into that organ. This would be true and affects our treatment of things in the brain, of course. It might be created in order to reduce the potential for inflammation and its damaging results. This would certainly be true in the retina. In the case of the thymus, realize that what the body is trying to learn is self versus non-self. So I do not want anything that is foreign to my body to be able to get into the thymus during a period of time when I'm trying to learn what the team uniform looks like. And I think you can imagine that if that were to happen, it would have catastrophic effects within the immune system because the cells that you're trying to train to recognize you would instead be learning to recognize you plus some other foreign invader. And this is one of many reasons why it is that having in utero infections and having microbial pathogens cross the placenta has such dire consequences for the fetus, because the blood to organ barrier in the thymus develops relatively late in gestation. And therefore, if I get that pathogen into the thymus when it should be teaching immune cells their eventual job, it's going to have dire consequences in the ability of that individual to recognize self versus non-self. Next, there have been rare genetic defects involving AIR and FESF2. And they have demonstrated in those patients that the lack of expression of those genes has a consequence in this process. Okay? Knowing what you know about those two transcription regulatory or transcription factors functioning in the thymus, how would you expect that patients would present if they had this genetic defect? Pause your video. Well, the name AIR, if you remember its acronym, gives you the answer to that. The name is shortened from autoimmune regulator. So if I cannot have the expression of all of the MHC molecules I need in the thymus, it means that my cells are going to leave the thymus with an inability to recognize some of those things as being self. So the outcome in the peripheral tissues would be autoimmunity. And these patients indeed have been shown to have dyscrasias of their ability to recognize their own tissues as self. Finally, a key component in the amyloid which damages the tissues of patients on long-term dialysis is beta-2 microglobulin. Can you explain why this is so? pause your video and take your time. Well, first of all, let's think about where beta-2 microglobulin is found. If you recall, it is a component of the MHC class I molecule. It's not encoded on the same chromosome, but it's simply added to the alpha chain to hold it in its appropriate shape. And if you move from there to the thought process of where is MHC class I expressed, you realize that it's on every single cell of your body with the exception of erythrocytes. So every time a cell in your body dies, and trust me, this happens many thousands of times a day, all of those molecules of MHC class I have to be degraded and processed through salvage pathways or whatever, and managed by the body either by excretion in the urine or the feces if those components cannot be reused. So when you think about the fact that the name beta-2 microglobulin tells you it's a very small molecule, you realize that our filtration systems that we use for dialysis have not reached the efficiency of the kidney itself. And therefore, we cannot remove that beta-2 microglobulin from the urine as it's formed, and it ends up being kept in the tissues and causing the production of amyloid. So this is a very important consideration on patients who have to be kept on long-term dialysis. And you should be able to walk that backwards to the molecular detail of where that molecule originated. Okay? So that ends this portion and ends the whole chapter involving the development of lymphoid cell receptors. So I'll see you next time.